Hello, everyone. Thanks for joining us for this webinar with Mahzi Therapeutics. We're really excited to have you join us for this. It's an incredible company, really doing the kind of work that gets overlooked a lot in the healthcare field, working with advocacy groups to promote cures for diseases that don't have the biggest patient population. So without further ado, I would like to turn it over to Yael Weiss, who is the founder and CEO of Mahzi Therapeutics, who will tell you about their unique approach. And again, if you do have questions, just type them in the Q&A at the bottom of the screen, and we'll get to those at the end of the presentation. So without further ado, Yael, I'd like you to take the presentation.
Thank you, Harvey. Thank you for the Force Family Offices for hosting us, and welcome everyone. I'd like to introduce you to Aaron Olson, our CFO, and myself, Yael Weiss, as Harvey said, the founder and CEO of Mahzi Therapeutics. I'm a physician scientist by education, but have been in the healthcare industry for many years, developing therapies on the clinical side, business development, and most recently as the CEO of Mahzi. Mahzi Therapeutics, and I'm happy to spend more time in the Q&A during the Q&A session talking about why and how Mahzi was founded, but we're fully dedicated to developing therapies for epilepsy, autism, and developmental disabilities. And I'll share my deck and start going through it. Just a sec. I want to start with some information that you may not be aware of, and just give you a flavor of why we decided to work on neurodevelopmental disorders. So 1 in 6 children in the United States have been diagnosed or will get a diagnosis of a neurodevelopmental disorder, and that's a huge number. These are numbers that are quoted by the CDC, the Center of Disease Control, and serves as a guide to us on the number of patients that are actually out there and we may be able to treat in the future. But what does it also mean? It means that you yourself may have someone, a loved one in your family that has a neurodevelopmental disorder. One of your neighbors may have a child with or an older patient with a neurodevelopmental disorder, families, friends. The issue with these disorders is because they're so severe, and the severe cases are cases that actually disable the families from being able to go out in public, to go shopping, to go to restaurants, to go on holidays. And these families are invisible. So many times you don't know that you may have a neighbor with a child with disabilities, but they are around you. And what Mahzi is trying to do is develop disease-modifying therapies that will enable these families to be more visible to the public, to go out, to have a quality of life that we all have, and enjoy the life as we do, and not have to stay at home with their child because they're not able to go outside. So why are we doing this now? We're doing this now because this is the time that neurology is actually shining. There are hundreds of new genes that are associated with neurodevelopmental disorders. And these neurodevelopmental disorders that we're working on are disorders that include epilepsy, autism, but profound autism, lack of verbal skills, that most of the kids cannot communicate verbally. They communicate with iPads if they can't communicate at all. They have developmental delays. A lot of them suffer from severe gastrointestinal symptoms and respiratory symptoms. So up until recently, if you had a child diagnosed with one of these disorders, you likely could not have gotten a genetic diagnosis because the genes were not known. If you look at the graph on the right-hand side, which depicts the discovery of epilepsy-related genes, you will see that if your child was born with epilepsy up till around 2010, you probably could not have gotten a genetic diagnosis. But fast forward to the 2020s, now all these genes are known. If a child seizes or has a developmental delay, they will almost automatically get a genetic test and get a genetic diagnosis if there is a genetic cause. And this means that the numbers of patients with genetic diagnosis will grow significantly in the next few years. and the years after. So we are hoping that by the time, or we actually know that by the time we will get to the market with these therapies, we will have a much larger commercial opportunity than what is thought to be had now. And we know also, and if you have been reading about the plausible mechanism that the FDA is talking about, we know that there is some regulatory acceptance of the uniqueness of rare genetic diseases. And this will lead, hopefully, to good discussions with regulators about approval pathways for these disorders. And we as a team, and we'll talk about the team a little later, we as a team have the unique capability to develop therapies for these disorders. So why us? Why Mahzi? We were built for purpose. We were built with the expertise to develop therapies for rare neurodevelopmental disorders. We have raised $60 million to date from blue chip venture capital firms, and we also received $8 million grant funding from the California Institute of Regenerative Medicine. We have limited competition because the commercial value is still hiding and hopefully will become visible in the next few years, where in most cases we will be the first company to develop disease-modifying therapies for these disorders. We have the development expertise. We've been running very efficiently. We're a small team that is very agile and very flexible, but very dedicated, and we've been able to achieve a lot with the funding that we raise. And we have just dosed our first patient in the clinical trial for a disorder called Pitt Hopkins, which I will talk about in a few minutes. And we're filing a second IND and going into IND-enabling studies with our third program, and all this in 4 years since the company has started. We are here because we're seeking additional funding to augment our clinical data package so that when we have a significant data package, hopefully by the end of the year, we will go out and raise our next round of financing. We're hoping to become a public company that specializes in rare neurodevelopmental disorders and be the leader in these disorders. We are already a go-to for many patient advocacy groups, and we hope to become even more so. A little bit about our partnerships with patient advocacy groups that Harvey mentioned. We realized very early on that parents are not waiting for pharmaceutical companies to develop therapies for their children. They are actually funding research. They're doing it proactively, raising money and bake sales, 5K runs, and providing resources to academics to develop therapies for their kids, whether it's a gene therapy, a gene editing tool, an antisense oligonucleotide, a repurposed drug. They're providing hundreds of thousands of dollars for research, but they get stuck. They don't know how to take these further. They don't know how to develop them in the clinic, and they usually don't have the resources to do so. So what we are doing is we're working very closely with patient advocacy groups, and you'll see in the middle some of the groups that we're working with, and we provide the knowledge on how to develop the drug so that when these drugs are available or ready with proof of concept, we can take them on and continue developing them. We provide resources, and we also allow these groups to invest in their programs. So not only will they have a therapy at the end of the process, but they will also have a chance to see a return on their investment if they did invest significant resources in their program. And Mahzi in Greek means together, and we wanted to depict the fact that we are working very closely with patient advocacy groups, working together with them, and that's the name. Just quickly, a quote from one of the patient advocacy groups that we're working with about the value that Mahzi brings to them and the power of working together as a company with a patient advocacy group. And we hear this over and over from the different patient advocacy groups that we work with. A little bit about Pitt Hopkins, which is our lead program, the lead disorder. It's a neurodevelopmental disorder that's caused by mutations in a transcription factor called TCF4. And it controls the expression of many other genes, which makes us hopeful that we can affect many cells in the body because so many proteins are affected by the mutation, or by the transcription regulation of TCF4, and mutation actually causes dysregulation of these pathways. The disorder is not inherited. The mutations are new. The parents are not carriers. Thus, the genetic diagnosis early on is extremely important, as I mentioned previously. And the clinical presentation of this disorder includes seizures, autism. Most of the patients are nonverbal and can communicate, as I said before, with an iPad. But some of them are so impacted, their motor skills are impacted that they can't even use an iPad to communicate. They also hypoventilate, and they have GI symptoms, very severe constipation that sometimes needs actually surgery in order to release the impacts. The AV gene therapy that we are developing for Pitt Hopkins is an AV9. On the left, you'll see the AV9 virus. Let me see if I can get the pointer to work. Probably can't. So on the left, you will see the AV9 virus that is actually a virus that we all have encountered in our life. But what we do is we degut the virus. We take out the genes that it needs in order to replicate within the cell and replace it with the genes that we want it to make. The virus then transduces, goes into the cell, into the nucleus. Its DNA is now where proteins are made, and the DNA actually replicates inside the nucleus, goes out into the cytoplasm, and makes TCF4 protein, which is the normal protein and not the mutated protein. And in this way, we augment the amount of protein that is needed and is lacking in the Pitt Hopkins patients. When we licensed the program, this is a program that was originally funded by the Pitt Hopkins Research Foundation. When we licensed it in, we had a very nice proof of concept that the gene therapy is correcting the mutation in brain organoids. Brain organoids are mini brains that are grown inside on a plate and are derived from patients or from their parent cells. So the parents serve as a control, and the patient's cells are actually the cells that we use to see if the gene therapy is working. On the left-hand side, you'll see that the parent brain organoids, the mini brains, have a very nice structure, and they have these green rosettes in there, which are the normal structure. If you look in the center, the Pitt Hopkins vector, the Pitt Hopkins cells have very few of these rosettes, and they're not as well organized. And when we gave the gene therapy, the organoids retained their normal structure and have a new formation of the rosettes, these green rosettes that are similar to the parental rosettes. We also saw improvement in EEG, electrophysiological defects that these organoids exhibited, and we were convinced that this is a gene therapy that could have potential in patients, and thus ran mouse studies in which we showed that in a mouse that is deficient of TCF4, very similar to what the patients exhibit, we were able to restore the structure of the neurons, which are these little trees that you see here. On the bottom, you can see that the dendritic spine length is normal in the wild type mice and the mice that do not carry the mutation. They're shorter in the mice that are deficient with TCF4. And when we give the gene therapy, we restore the length of these neuronal dendrites back to the normal size. So, and we also were able to show, and we don't have the graphics here, but happy to share them. We were able to show restoration of mouse behavior. Several behaviors that are autistic-like behaviors were restored and back to normal, and we were able to show this in the dose-responsive manner to show that and show the dose that we're planning to use in the clinic is effective in mice as well. Where we are with this program is what we've had many interactions with various regulatory agencies around the world. We have an active IND in the United States. As I mentioned, we dosed the first patient last month. We have sites activated in the US, in San Francisco, Denver, and Chicago, and we're waiting to open sites in Israel and in Spain. and we are hoping to see early data readouts by the end of the year. This is our pipeline. On top of the TCF4 program, which I just presented to you, we have another gene therapy that we will be filing an IND for later this year for a disorder called WOREE. And then a pipeline of antisense oligonucleotides. We are modality agnostic. We develop gene therapies and antisense oligonucleotides for 3 additional disorders: CHD2, SCN2A, and ARID1B. Aaron, I'll hand it over to you to talk about the pipeline strategy and our progress to date.
Thanks, Yael. As Yael mentioned, our lead program is an AAV9 gene therapy for Pitt Hopkins syndrome. It's one of the largest genetic neurodevelopmental disorders with an estimated 30,000 patients in major commercial markets. We forecast a peak annual revenue opportunity of approximately $500 million. MC1866 is the first investigational therapy to address the underlying biology of the disease, and we've just recently initiated the phase 1/2 clinical trial. Our second program is also an AAV9 gene therapy for a devastating epilepsy disorder called WOREE syndrome, characterized by profound developmental delays and early mortality. Although a less prevalent condition than Pitt Hopkins, with an estimated 8,000 patients, we have strong non-clinical proof of concept data, and we may be able to accelerate clinical development and approval with recent FDA guidance on expediting treatments for rare diseases. We plan to file an IND for this program in the first half of this year. We also have a robust portfolio of early-stage antisense oligonucleotide, or ASO programs, and indications with prevalences that average 20,000 patients or more, and with peak annual revenue opportunities that range from $200 to $600 million. As Yael mentioned, we plan to advance at least one of these programs to IND-enabling studies this year. And now I'll pass it back to Yael for a bit more background on our company.
So thanks, Aaron. And this is our team. I will not go through them one by one, but you will see that we all have significant experience working in big pharma and biotech companies that specialize in rare diseases such as Ultragenyx and BioMarin. And this is our board and our advisors. I'll highlight the fact that John Marganorig, who you may have heard about, is our advisor. He was the founder and CEO of Alnylam Therapeutics. And also on the bottom right, Janet Woodcock is our senior advisor. She was the former director of CDER and deputy director of the FDA. And we are the only commercial company that she has agreed to consult for because our mission resonated with her. So I'd like to summarize and remind you that we're a clinical stage company. We're targeting underdiagnosed genetic neurodevelopmental disorders, hoping to have thousands of patients diagnosed by the time we get to the market. We're driven by gene-specific biology and clinical expertise. We are first mover in many of these disorders, which is especially important in gene therapies to capture a large number of patients. We work closely with patient advocacy groups. We are modality agnostic, gene therapy, and antisense oligonucleotides. We raised $60 million to date and $8 million in non-dilutive funding. And we're looking for an additional $10 million that will bridge us to a full clinical data package that will lead to Series B fundraising and IPO. Thank you very much, and happy to answer any questions.
Great. Super. We got a bunch of questions that came in, so why don't I go ahead and get these to you, and you guys can answer them for the audience. First one. So is Mahzi primarily a pipeline company or a platform company? And how many assets do you believe can be generated in the next 5 to 10 years?
So I think the answer to this question is that our platform is our expertise in developing therapies for neurodevelopmental disorders. However, because we have this expertise, we're able to bring in the most suitable assets for each disorder and thus become a pipeline company. And we are spanning AAV gene therapy, antisense oligonucleotides. We're looking into gene editing programs and also small molecules. So we can pick up any therapeutic modality and take it into the clinic, and this will become our pipeline. We're hoping to have, and based on our interactions with patient advocacy groups, at the moment we have 10 relationships, at least, at minimum, I think, with patient advocacy groups. helping them develop therapies for their disorders, mostly antisense oligonucleotides, some AAV9 programs, some editing programs that we could potentially bring into Mahzi if these programs reach proof of concept and they're successful and we have the funding to do so.
Great. You mentioned you've raised $68 million so far. What is the current cash runway and annual burn? And you've already answered this, but we'll do it again. How much are you looking to raise right now?
I can answer those. So annual burn is about $12 million per year. So again, very small, lean company with 14 full-time employees. Runway is through Q3 or September. And we are looking to raise $10 million now with purpose of getting through a full clinical data package for our lead indication, as Yael mentioned.
Great. Your preferred exit strategy seems to be an IPO. What do you foresee as the time horizon for that?
So I think a lot will depend on the quality of the data that is generated from our first clinical trial. But if the data looks as good as we're hoping it will be based on our preclinical studies, then by the end of the year, we may be able to raise either a Series B or a mezzanine round that will take us into an IPO in the next few years.
Great. For the Pitt Hopkins program, what are the key clinical endpoints and what would constitute proof of concept in phase 1/2?
So it's a question that obviously we spent a lot of time thinking on. The phase 1/2 study, to remind you, the main endpoint is safety. However, we have multiple exploratory and secondary endpoints that will be used to define what an approval package would look like. We are using measurements of psychometric measurements, epilepsy. So we have seizure tracking. We also include tracking of improvement in GI symptoms, respiratory symptoms, and motor and cognitive skills, obviously. We're following the patients with videos. So starting from baseline throughout the first year of the study, we will be able to capture any improvements by video, which I think would be very telling if there is significant improvement in this patient population.
Right. So what is the biggest scientific unknown for Pitt Hopkins today and like vector delivery, dosing, or durability?
So I think based on what we're seeing from other companies that are developing AAV gene therapies, we are seeing efficacy in disorders like Rett syndrome, Angelman, and Dravet. Obviously, the question of durability is a great question for AAV9 in general, or any gene therapy. We will know in coming years. We assume that in the brain, the treatments are durable because the neurons do not divide, and thus the effect will not be diluted, but we don't have the data to show that. Regarding vector delivery, we know that the AAV vector does not transduce a large percentage of the cells in the brain. Only part of the brain is transduced by the virus. However, because this is a transcription factor, and we know that it actually oversees the synthesis of many proteins that are secreted from the cell where the virus is. The proteins will be secreted and will be affecting other cells. We're hoping to see cross-correction in other cells in the brain that did not necessarily get the virus.
Right. Do you envision one-time gene therapy pricing, outcome-based reimbursement, or annuity-style payments?
At this point, we do model one-time pricing. Outcome-based pricing is possible. We don't think right now that we'll commercialize on an annuity model.
Okay. How do you think about dilution versus partnering? What inflection point would trigger a pharma partnership or licensing deal?
So at the moment, we've had actually some inbound interest from Japanese companies for regional deals. And I think if we do decide to partner sooner better than later, it will be a regional type of partnership. I don't know that we would fully license or sell the program to anyone else until we have additional programs in the pipeline that we know are strong enough to maintain and lead the company forward. For US-based pharma at the moment, gene therapy is not the flavor of the month. For mid-sized companies, there is still interest in gene therapy. But as I said, I don't think we will fully partner the program, but we'll look for a regional partner, whether it's in Asia/Japan or in Europe, and not give out all the rights to the program.
Got it. What preclinical data gives you the highest confidence that gene therapy and neurodevelopmental disorders will translate to meaningful functional outcomes?
So it differs from one disorder to the other. For Pitt Hopkins, I showed the mini brain, the brain organoid, and the mouse model. Both showed improvements and good response to the gene therapies. For our second disorder, WOREE, the mouse data was very compelling. Mice that normally die with a mutation at 3 weeks of age, lived and are able to reproduce and restore all the functions of a normal mouse. So obviously, knowing that these are mice, the clinical data will be the one that will speak for the efficacy of the program. But we make sure that we bring in programs that have solid proof of concept, at least with the available tools, which are either in vitro or in vivo tools, and advance them into the clinic. For antisense oligonucleotides, there's a lot more history in neurodevelopmental disorders, starting from SMA, Angelman, and Dravet. So we know that in most cases, these preclinical packages translate pretty favorably into clinical outcomes.
Great. Can you talk more about the relationship between the company and the patient advocacy groups? What does it mean to be helping these programs develop?
So when you receive a diagnosis of a genetic disorder, you likely were not a professor in biology or a major in biology, and usually not a drug developer. So a lot of these groups organize, fundraise, and Google their gene, find the lab that's working on their gene and fund it. What we're trying to do is change this paradigm and educate these patient advocacy groups not to necessarily go to the first group that they found on a Google search, but put together a drug development team from the get-go. And for each group that we work with, we help them identify the best modality for their gene or their disorder. We work with them to identify the best, for example, the best sequence for an antisense oligonucleotide to tackle, or the best constructs and AV constructs to design for their gene, so that and help them design the studies that will prove that the therapy is actually working. And then once this proof of concept data is available, we have an option to license it. And if we are in a position to in-license it from a resources and timing perspective, we will in-license the program and continue developing it. If not, we grant these advocacy groups the right to partner it with someone else who will help them develop it.
Right.
I hope this answers the question, but it's a very, very close and tight relationship. We also help them get ready for clinical studies by having natural history studies, a registry, a disease concept model, making sure that the patients are educated on what the gene therapy or the antisense oligonucleotide therapy could potentially do, the benefits of these modalities, the risks of these modalities, so that when time comes and we're ready to recruit for a clinical trial, the community is ready to join us. So, for example, for the Pitt Hopkins study, we've been working with the community for the last few years. We have a wait list at any given moment to enroll in the trial, and we don't have to go looking for patients, which will slow down the enrollment into the study.
Right. The FDA put a clinical hold on an AAV9 vector product a few weeks ago, suspicion it caused a brain tumor. Will this affect your dosing of additional patients, or are potential subjects in your clinical trial concerned?
So for the specific program that you're asking about, Frank, the construct and the promoter are very specific. So let me take a step back and say there was always known that there was a risk of AV integrating. We know that it happens at a very, very small percentage. Now, the combination of the vector and the promoter that that specific company used, a very strong promoter that causes expression of the gene to be above normal levels, probably contributed to this. I don't know. I'm speculating. I obviously don't have all the information. In our case, specifically for Pitt Hopkins, for TCF4, we're actually using the gene's own regulatory elements. And we know that it does not overexpress. We actually have physiological levels of expression. So we feel comfortable about the levels of expression and the risk. But every patient that enters our study in the informed consent forums acknowledges the fact that there may be a risk of cancer in the future. I would like to emphasize that these families, the need is so significant that they are willing to take the risk of a future cancer for the potential of improvement of their child's life and the family's life.
Great. Thank you. What are the steps for a patient advocacy group to become considered for the Mahzi pipeline, and are ultra-rare conditions under 100 too small?
So our smallest disorder is WOREE, which is our second pipeline program, and there are about 300 patients currently known to be affected by this disorder. The criteria that we employ is a strong patient advocacy group, a strong proof of concept package, and the potential to identify additional patients using genetic testing. As I said before, I'm very hopeful that the surge in genetic tests will lead to an increase in populations of all these ultra-rare disorders. And if we can see that there is a trajectory of growth in the patient numbers over years, then we will consider bringing this program on.
Great. So congratulations on the progress to date. In terms of your next clinical steps, you've mentioned the US and Spain. Have you considered international sites such as Argentina with an active Pitt Hopkins Patient Association group and well-defined centers of excellence?
So our first study is very small. We're enrolling only 12 patients, 2 cohorts of 6 patients each. So with the number of sites that we currently have, 3 in the US, 1 in Spain, and 1 in Israel, I think we can enroll the study by the end of the year. As Aaron mentioned, we're a very small company and we run a very lean organization, and opening additional sites would be costly, and we're trying to balance spending and successful enrollment into the study. If we do need to increase the number of patients and enroll additional patients in order to get approval, then we will definitely consider opening additional sites.
Great. Well, that concludes all the questions from the audience. Thank you so much for those questions. If you would like to follow up with the folks at Mahzi, with Yael and Aaron, please get in touch with us and we'll pass your information on to them. Conversely, we will give your contact information to them and they can be reaching out shortly. I would now like to turn it back over to Yael for any final words before we wrap up.
Thank you, Harvey, and thank you for everyone that participated, and thank you for the great questions. We're very passionate about getting treatments to neurodevelopmental disorders and their patients and their families, and doing this in a way that would bring value to investors, to patients, and to their families. So I hope you'll join us on this journey. It's an exciting one. And as Harvey said in the beginning, an underestimated market that we hope to prove everyone that we are stepping in first, being the first ones to capture a lot of these diseases, and that we will be the first one to deliver a therapy to these patients, and other companies will follow us. So thank you very, very much.
Great. Thanks so much, everyone, for joining, and everybody have a great day.